And thank you all for being here today to participate and for your participation at the SDB as well as this awards session. It gives me absolutely great pleasure to be able to bestow on behalf of the Society for Developmental Biology the Lifetime Achievement Award to our friend and colleague, Dr. Eddie de Robertis. I would like to share my screen presently so I can show you something very quickly. Hopefully you can see this. So this is what the actual trophy looks like. I wish I could give you this in person, Eddie, but you will be getting this in the mail at some point, I presume. But I just want to provide you with a little bit of a story and history, perhaps, of what it's like to be a member of the Society for Developmental Biology. I have to say that I first met Eddie when I was a undergraduate student in 1981, but I did not meet him in person. I just didn't know that I was going to meet him in person, but I met him through this particular book. It's a book that he and his father wrote. This is probably the 10th edition. This is my own copy from 1981. And it was an introduction to me in Spanish to the fields of molecular and cellular biology. I have to remember, this is in Venezuela, 1981. I spoke no English. So this was kind of like my window into understanding the complexities and the inherent beauty that there is in studying the molecular and cellular basis of temporal transformations of tissues and cells and all. And so it's remarkable when I think about it that today, I would be actually presenting this award to Eddie. And it's hard to imagine in 1981 that that would be happening today in 2021. But let me tell you a little bit about Eddie. Eddie was born in Cambridge, Massachusetts. He was born there because his father was doing postdoctoral training at MIT. And then after his father concluded his work at MIT, they moved back to South America. Eddie received his MD and PhD from Uruguay as well as Argentina from the Leloir Institute. And he did postdoctoral training with John Gurdon, as well as moved from John Gurdon's lab to Basel to take hold of his first independent position at the University of Basel. The one thing that I want to bring to people's attention, because this is something that is usually not readily associated with Eddie, is that I believe that Eddie's work in 1984 along with his colleague at the time, the late Walton Gehring, of isolating the first vertebrate developmentally controlled gene, which we now call HOXC6, was really probably one of the most influential findings in embryology at the time. It was because it helped provide, for the first time, I think, unambiguous evidence that evolutionarily conserved mechanisms were actually controlling embryonic patterning across a broad number of organisms out there. So in short, I think Eddie's work has shown that the patterning of all known bilaterian organisms occurs via conserved developmental pathways. This is something that we now take for granted, but it's something that wasn't entirely certain just a decade and a half ago, that it would actually be the case today. Beyond being an extraordinary scientist, Eddie has also been a terrific mentor to both members of his lab as well as members outside of his lab, particularly many of us in the developmental biology community. And with that, Eddie, I just want to congratulate you and wish you the very best, and very much looking forward to your presentation. Thank you.
Thank you very much. Let me share my screen. Thank you so much, Alejandro. And I would like to thank, the SDB for this lifetime award, which means more to me, more than you can even imagine, because this is my society. This is what I have done all my life. So it validates a life with the embryo. So I will tell you what got me here, just first from the Hox genes to Sprague organizer, But then how the importance of the cordon gradient, how does that tell us about the embryo regeneration and especially about evo-devo? And I would like to end in some unpublished work that relate lysosomes to the early Wnt signal. In our view, Wnt is a cell biological signal of great importance. And to get here, you first need to have the best mentor possible, which I did. Most of you know John Gurdon. And after 6 years with him, at age 33, I was appointed full professor in Basel, Switzerland, where I completed really my education with my colleague, Walter Gehring, just one of the greatest developmental biologists. And there we did this important experiment, which was just mentioned, the first Hox gene cloned from a vertebrate in a collaboration. And at the time, no one imagined that a fly would develop similarly to a vertebrate. So it was a wonderful moment. And that gave me more lift. And I got an offer from the University of California in Los Angeles, which for an endowed chair. And so in 1985, we moved to Los Angeles, and we're still there in the same place. And there our work took a different turn. We studied the most famous experiment in embryology, which is the experiment done by Spemann and Mangold in 1924. And what they did, here repeated by me, what they did was to transplant the dorsal lip of the blastopore, which is this region where gastrulation starts, and transplant it into the ventral side of a recipient embryo. I show this movie because I learned how to do this procedure from a movie. So I think that maybe other people can learn it too. So I keep on showing it. You can do this freehand, and you take a hole in the embryo and you put your graft within it. Then you give it a little push and a push. And within an hour, the embryo heals almost miraculously. And 2 days later, you get an embryo which has a second axis, in which the transplant gives the notochord mostly, but the brain, the eye, the cement gland, the somites are all induced in the neighboring tissue. So this was the discovery in 1924 of embryonic induction. And Spemann received the Nobel Prize in 1935 for this. But there was nothing much that could be done until in the 90s, molecular biology became practical, and a number of groups tried to look at what were the genes that had this incredible inductive activity. And we first isolated a homeobox gene that marked the organizer, and then many secreted genes were isolated. These are BMP antagonists. This is a nodal antagonist. All of these are Wnt antagonists, of which the one that we're going to talk today is cordon, which is at the nerve center of the Spemann organizer. It is expressed in regions which, when transplanted, have activity as organizer. When you deplete this gene, the organizer loses its inductive activity. And this is the RNA, but the protein diffuses in the embryo where it acts as an antagonist of BMPs. And this gene was cloned by Yoshiki Sasai, who gave me cordon, and is the same person that gave all of you organoids. So he was a great developmental biologist. And as the years went on, we realized that it was not just cordon, but also there were other genes in the opposite pole of the embryo that were part of a network of interacting extracellular proteins, like this one called sizzles. And this network has on the dorsal side BMPs that are made dorsally. Together with cordon, they're neutralized, they flow to the ventral, where they're then cleaved by a metalloproteinase called tolloid, and tolloid will cleave this complex, release active BMP, get to peak BMP signaling, and then allow the transcription of more tolloid and the transcription of sizzled, which turns out is an inhibitor of tolloid. So for in a cartoon shape, you see that cordon is a large protein with BMP binding modules. It binds BMP, and tolloid is this little scissors that cut them in two in the particular places, which we identified. And together with a cofactor called twisted gastrulation, BMP can then diffuse and bind and signal through its receptors. But on the ventral side, tolloid might be confronted with sizzled, and it will recognize sizzled and will try to cut it, but it cannot cut it. So sizzled is a classical enzyme competitive inhibitor for tolloid. And from this cycle, then you get this constant gradient formation of BMP. We have been able to visualize the cordon gradient from the dorsal of the embryo. It forms the protein, now with an antibody, to all the way to the ventral. This is a very long way, in this case, 2 millimeters from here to here. And most of the cordon is found in a narrow extracellular matrix or signaling highway that separates the ectoderm from the mesoderm, called Brachet's cleft in Xanopus, and also the endoderm. So from here, you can create a gradient that will pattern both the ectoderm and the mesoderm, so that you get coordinate development of the germ layers in the embryo with respect to a gradient of BMP signaling, which is the reverse of the cordon gradient. And we've done experiments to show that all this is driven by the degradation of cordon by tolloid, which is the key part of this. Now, understanding this gradient of cordon has many advantages. It helped us understand, for example, a very old experiment in which the frog embryo is less pigmented on the dorsal than on the ventral, but you can take a blastula and cut it in half before the organizer is formed. And from this half embryo, you're going to get the ultimate regeneration. You're going to regenerate from half the whole of the missing half. In the other case, also the whole of the missing half. Both come from the same egg, these two. So how can that be that you get this amazing regeneration? When you cut the embryo, you create this enormous wound in the embryo, and Yuki Moriyama found that what happens is there's a healing so that the dorsal side, less pigmented, will come to lie next to the old ventral side, and then from the opposition of this high ventral with this high dorsal, the organizer will be formed at a 90 degrees displacement. So instead, it's going to be displaced, and there the dorsal lip is going to involute, so that instead of forming in this regular region where you would have expected, really it goes 90 degrees, then involutes, and then creates the right side and the left side of the embryo, with the high BMP region on the opposite side of where cordon is formed. So this is published, and it's quite interesting. How do you make a twin out of a single egg? And also, knowing cordon allows us to understand things about evolution, probably. So Darcy Thompson, in a classical book 100 years ago, wondered, why does the pufferfish, which is in fact a close relative of the sunfish, why do their body shapes look so different? There must be some underlying mechanism. So here, to celebrate his centennial, I did this experiment of injecting sizzled morpholino only on the ventral side of the embryo, which is more pigmented. This is the less pigmented. So if you decrease sizzled, you're going to get more tolloid, you're going to get less cordon, you're going to get more BMP, you get more BMP, you're going to get more ventral posterior tissue. And since we only injected the ventral side, the head remains the same. So the head is normal, the posterior ventral is expanded. So you could argue, well, maybe it's a change like this. You could say, oh, that is maybe, you know, an exaggeration, but it turns out the experiment has been done by nature, and Kinya Ota in Taiwan has found that, you know, the twin-tailed goldfish, which has been kept for hundreds of years by aquaculture, that it's a very old mutation. I had them when I was a child in a fishbowl. They've been kept because the cordon mutants, they like to swim in a fishbowl. But if I want you to see that the shape of the body is completely different from one goldfish to the other. So then this tells you that cordon is a morphogenetic protein that makes morphology. So makes morphology of the adult body plan and of the embryo. And this turned out to be quite important because what we found in the frog about cordon and toloid and BMP has been found also to be true in many other organisms. For example, in the fruit fly, where cordon is called SOG and BMP is called DPP. And then in many animals, but interestingly, even in the cnidarians, Technow has found that during the formation of the blastopore in the sea anemone, you have a patch of cordon and DPP. This diffuses through the embryo where toloid will cut the cordon and will release so that there is a gradient of BMP with the maximal opposing the place of where you have cordon. Therefore, in the vast majority, not all, but the vast majority of the bilateral animals, the cordon toloid BMP pathway is a molecular machine used to establish a diffusion gradient that spans the entire animal embryo. So this is an ancient patterning system. And in 1996, a long time ago for most of you, we got the good idea with Sasai to write a review in Nature where we proposed that the primeval bilateral animal, Urbilateria, was an animal of great genetic complexity that gave rise to all the protostomes and all the deuterostomes, and it contained the cordon toloid BMP, and it contained the Hox genes in the anterior-posterior axis. So what I would like to tell you today is not only that this animal living in the bottom of the sea was complex, it also had a planktonic larval form. Shown here is a larva for an annelid, which is characterized by two cilia rows that beat in opposing directions, an apical tuft and an ocellus, a little eye. So this would be like for an annelid, but the similar larvae are also seen in hemichordates and in sea cucumbers that are deuterostomes. So probably this was had a complex life cycle, in the bottom of the sea, and how unlikely is that from this creature all the animals came through evolutionary changes in these development control genes that are still used today. So with that, I would now like to tell you about unpublished work that we're doing in the wind signal, because cordon is turned on by earlier gradients, of which the first one is this gradient by wind. And what we found a number of years ago is that during wind signaling, this is wind through its receptors, it's very important for the receptor complex that contains an enzyme called GSK3 and an adapter called axin to become sequestered inside multivesicular bodies. So now, as part of the normal membrane trafficking, you have this outside-inside vesicle formation by these escort proteins. It produces the sequestration of active GSK3 from the cytosol into these organelles, and this causes protein stabilization by decreasing cytosolic phosphorylation of phosphodigrons that normally are required for protein degradation. The most famous one is beta-catenin, which becomes stabilized and explains all the transcriptional activities of Wnt. But there is much more to GSK3 than just, well, so that the inhibition of GSK3 than just beta-catenin, because 20% of the human proteome we found has 3 or more GSK3 sites in a row. And this explains the stabilization of many proteins in a process that is now called Wnt stop, and it's part of the Wnt field. So what we found more recently in these 2 papers here is that when Wnt signals, it triggers a process called macropinocytosis. Usually it used to be called pinocytosis or cell drinking. It triggers the local inhibition of GSK3, triggers the formation of actin lamellipodia that engulf microns, vesicles that are microns in size, that will sequester the many proteins from the outside and take the receptors into the multivesicular bodies and into the lysosomes, so that now proteins that are on the outside will go into the lysosome. There will be very important and big nutritional changes. So I'll illustrate that with the uptake of bovine serum albumin into a group of cells. These are 3T3 cells, and they're put with a bovine serum albumin for 30 minutes. And you see that without Wnt, there is very little uptake, but with Wnt, the BSA is taking up. And this is not just any old BSA. This is cleaved, because this is a derivative which fluoresces when it is cleaved in the lysosome. So these are active lysosomes, and we've shown that the lysosomes, when you put Wnt, become much more acid. The lysosomal proteases, such as cathepsin D, become activated, and there are big metabolic changes in the numbers, the types of amino acids that then are digested from these lysosomes. And importantly, all these changes don't need any transcription, or no translation of new proteins, because they take place in cyclohexamide and they take place almost instantaneously. So in our view, Wnt is a regulator of membrane trafficking, and that's a very important part of its biology. It's not all beta-catenin. So this, now what about the embryo? And so here are 64 cell embryos stained with lysosome, which is a reagent that stains the active form of cathepsin D. So you can barely see that there is an enrichment on the dorsal side of active lysosomes as opposed to inactive lysosomes. But if we inject Wnt8 mRNA into the vegetal pole of the embryo, you see the dorsal side, which is less pigmented. The dorsal side has a large increase in the amount of active lysosomes. This embryo will be then dorsalized if you let it grow. And in fact, if we inject lithium chloride is a Wnt mimic because it's an inhibitor of GSK3. And then you see that then that the lithium chloride also produces a large increase in GSK, in lysosomal activity. So we think that at these early stages of development, this formation of lysosomes, this is an activation of lysosomes, is very important in the Wnt signaling and in the embryonic patterning. So this reminded us that now I'm going to tell you about the work of Nidia Tejeda Munoz. It's all, as was the previous slide, that we knew that there's chloroquine is a drug that alkalinizes, weak base alkalinizes lysosomes and blocks Wnt at high concentrations. But at low concentrations, it allows the formation of these intraluminal vesicles of the MBVs. Each of these little vesicles has been formed by the escort machinery you see here. And so it turns out that at low chloroquine concentrations, Wnt can be 2 to 3 times more active. But there's this topical drug now called hydroxychloroquine, and Nidia found that is much more effective than chloroquine at inducing activity of beta-catenin in a Wnt assay in HeLa cells. So this is with Wnt, but Wnt plus hydroxychloroquine, it goes up, in this case, at this concentration, 34 times. And this, of course, requires Wnt, because if you put hydroxychloroquine alone, it doesn't have any effect, the hydroxychloroquine. And we do it, if you do it with lithium, it's the same thing. It increases, although here only by 6.5%, but hydroxychloroquine alone doesn't do anything. And in the embryo, this effect of hydroxychloroquine on increasing Wnt signaling is quite spectacular, because we can inject 4 nanoliters of lithium chloride at 300 millimolar into the ventral side of the embryo, and you can see that you get a slightly dorsalized embryo, big cement gland, big eye, big head. But if we put hydroxychloroquine between 1 and 10 millimolar, and it will form a radial embryo with a radial cement gland, a radial eye, a radial blastopore. This means that it has a radial organizer. And this requires this process, for example, of macropinocytosis. Well, there is an inhibitor of macropinocytosis called IPA. So if we co-inject lithium hydroxychloroquine and inhibit macropinocytosis, all this goes away, and if I have a regular embryo, and IPA alone or hydroxychloroquine alone is without effect. We can get the same things if we block the formation of multivesicular bodies with HRS morpholino. So then what we have is then first that hydroxychloroquine might be working through the increase of Wnt, not through the inhibition of the lysosome. But more importantly, we feel that the lysosomes early on in development must be very important in the early Wnt beta-catenin signal, which is the dorsal determinant in the frog, which is so important. And I expect to devote the rest of my scientific life to try to study this cell biological properties of the egg and their lysosomes. With that, I come to the end of my talk, and I would like to thank the frog embryo, which is so good to me, and my many colleagues throughout the years that have been also so good to me, and my present group, people that are working with me now. And I will leave you with these thoughts, but in particular, what I want to do is to thank again the Society of Developmental Biology for this extraordinary recognition. I am very happy to have received it. Thank you, my peers, so much.